So osmosis is, that means, so movement of the water, these are the water molecules. This is the movement of the water molecules across the cell. So movement of the water across the cell membrane, down its concentration gradient. So down the concentration gradient. As we know, the water is hydrophilic substance, right? The water is hydrophilic substance. It's hydrophilic. So it passes through which diffusion if it's hydrophilic? Down the concentration gradient. If it's down the concentration gradient and the substance is hydrophilic, that means it is, most possibly, it is facilitated diffusion. How many ATP molecules are they going to use?
18, right?
Saudat, what do you think? How many ATP is used to move the water molecules inside the cell or outside the cell?
No, 2, right? No.
Okay, Nurhayat is saying 2. Saudat, what do you think? You have to unmute yourself.
I don't know.
Guess, just guess.
Maybe 5.
5. Gulkhaya, what do you think? Munisa?
A lot? Like
Ella?
more than 50?
More than 50,
More than
okay.
50 or 1,000?
Okay, it's facilitated diffusion. We are saying it's facilitated diffusion. Facilitated diffusion. In the last lecture, we learned that the facilitated diffusion, right? It is also type of the passive transport. It is a type of the passive transport. And no energy is required. Why you are saying ATP molecules 5, more than 50, 2? Why? We learned about this. No energy is required if it's facilitated diffusion. You should say only 0. 0 ATP is used, okay? Do not be confused. We learned this information, okay? And the types of the osmosis also divides into 3. We have 3 types of the osmosis. The first is hypertonic, second is hypotonic. This is the first, second, first, and the third. Third is isotonic solutions. We can substances will move. Is it from me? Is it coming from? Or can you hear me? Can you hear me?
Yes.
Okay, so these are the solutions. Have you ever heard these solutions somewhere? Have you ever heard this? No? Okay. So let's start. So what's hypertonic solution? What's isotonic solution? Let's break it down. So first of all, let's take the glass of water. Actually, what did this look like? What is it? What do you think these reddish structures? What are they? What are they?
Hemoglobins?
Like tailets?
Yes, they hemoglobin molecules. They are red blood cells, right? They are red blood cells, hemoglobin. Let's take a glass of water. This is the glass of water. This is also glass of water. And this is also glass of water. Let's imagine, okay? They are glasses of water. So in this glass, we have the water, water, and water, okay? So we have the water molecules here. What's going to happen in the hypertonic solution? What's the meaning of the hyper?
It's a lack of water.
Yes, this is means high, right? Hyper means high or a lot. And the tonic means what?
Li liquid.
What's the meaning of tonic? Tonic means salt. Salt, okay? What's inside of the salt? What elements inside of the salt?
Potassium and no, sodium and
Chlorine, right? Sodium and
yes.
chlorine. The main focus is, our main focus is sodium today. So imagine hypertonic solution. Solution is more related to the water molecules. So if you say solution, so we understand the water molecule. That means a lot of salt in water. Okay, so we have a lot of salt molecules, potassium ions in the water. Okay, a lot of salt. Hypertonic solution means the less salt compared to the hemoglobin. Okay, we have the less salt compared to the hemoglobin. Isotonic solution means the amount of the salt and the amount of the salt in the solution is equal. Okay, isotonic means iso means equal. Okay, hypo means low, tonic means salt, so that means low salt solution. Okay, so what happens in the hypertonic solution? Imagine I have the red blood cell. This is called the hemoglobin. In this hemoglobin molecule inside, we have the ions as well, and we have the water as well in this hemoglobin molecule. Okay, water as well. So if I place the hemoglobin, imagine I have the hemoglobin, and if I place this hemoglobin into this hypertonic solution, what's going to happen? What do you see in this image?
Lack of water. Like
Yes, the water inside of the hemoglobin is going to release outside. They're going to release into the water itself, and the hemoglobin is losing water. Hemoglobin losing the water molecules. Okay, and they are shrinking. This process is called shrinking. Shrinking is when a substance loses water and they become shrink. Okay, this shrink, they're going to shrink. And my question is, why the water molecules in hemoglobin? The water is leaving the hemoglobin. Why?
because there's a big amount of salt.
Yes, because it is hypertonic solution. In the water here, we have a lot of salt molecules, right? A lot of salt molecules. That means they are attracting the water. What is salt? There will be the water. So salt is going to attract the water molecules. They are going to attract, and that's why it shrinks. For example, have you ever made salonni yopganmisizlar? Qilganmisizlar salonni? Kim qilgan salonni? Qo'lini ko'tarsin. Gulhayo salonni qanaqa qilishadi? Gulhayo, we are waiting to you. Now, so we get the can, right? We get the can. This can, we add the water or we add the cucumber and salt, right, to that water. We add the salt to that water. We add the salt. So what happens to the cucumber then, Nurhayo?
It absorbs the salt.
It doesn't absorb the salt. It gives off the water from itself. It shrinks. Number is going to shrink. It's going to shrink. It's going to become smaller. Okay? It's going to shrink. It's going to get smaller. Okay? In shape. It's going to give off the water. It gives off the water outside. Okay? It gives off the water. That means they are losing the water because the salty, because the water contains the salt. This is the example to the hypertonic solution, okay? So in the hypertonic solution, imagine we have the less salt molecules here. We have the less salt in here compared to the inside of the salt, okay? It's very less. So what's going to happen here? We have the sodium ions inside. We have the high sodium amount inside and low sodium amount inside the solution. So what's going to happen? And a lot of water we have around, a lot of water molecules around. So these water molecules is going to go where the salt is a lot, whereas the sodium is a lot, okay? In this type of the solution, the hemoglobin molecule, this is the hemoglobin molecule, this is the red blood cell, this is the hemoglobin. So this hemoglobin is going to increase in size because water is moving inside of the hemoglobin. This water is entering to the hemoglobin, okay? Because we have higher amount of the sodium in hypertonic solution. That's why it is swelling. We call this process what? We call this process swelling, okay? And it's bursting. It is bursting. We call it bursting. Okay, so it's going to rupture, it's going to burst, and we lose this hemoglobin molecule if we put it to hypertonic solution. In the isotonic solution, this is the solution which contains 0.9% of the sodium, okay? 0.9% of the of sodium. That means in the hypertonic solution, we have more than 0.9% of the sodium. In hypertonic solution, that means they have the salt molecules or they have the sodium which is less than 0.9, okay? This is how we differentiate the 0.9. This is the standard, okay? Have you ever seen the sodium chloride 0.9% and it's written isotonic where you inject your veins? Have you ever seen? This is the isotonic. It is 0.9%. So your cells will be normal. There is a normal exchange of the water and sodium. It will be normal. Your hemoglobin won't be, your cells won't be shrinked or won't be swelled in this case. Okay, they won't be shrink, they won't be swelled. This is the isotonic solution. If you have I gotta remove the water to the brain. Okay, do you understand me?
Yes.
Is it possible? Okay, so in the hypertonic solution, imagine if you have the shrinkage of the cells, you give the hypertonic solution so that they're going to swell. They're going to, so it depends on the situations you give any solutions, and these examples to the osmosis. So you have to learn this skill to identify which solution does what. In hypertonic solution, the cell is swelling and bursting. In isotonic solution, nothing is happening. No change in the hypertonic solution, no any change in isotonic solution. And in the hypertonic solution, the cell is going to shrink. Okay, or we can call it shrivel. Okay, we also can call shrivel and shrink. They are the synonyms. And no change in the isotonic solution, and swelling of the cell, swelling and bursting in hypertonic solution. Okay, any questions regarding this? Any questions?
No.
Okay, cytoplasm. What's cytoplasm in the cell? What's its function? Who can answer? What's cytoplasm and its function?
It's like a jelly which contains the organelles.
It's a jelly-like semi-liquid content which contain on itself what? Which contain on itself the organelles, right? Organelles. What are the organelles?
Vacuole. Something.
Yes, we will learn all the organelles. So you're going to answer all of the organelles you will learn by heart. Okay, so cytoplasm, this is the semi-liquid. As you can see here, this is what structure is this?
Structure of the cell.
Which structure I am showing now?
Cell membrane.
Yes, it's cell membrane. If you go deep to the cell membrane, you see the lightish bluish color, right? And lightish bluish color, this is the cytoplasm. Okay, this is the cytoplasm. And everything colorful you see in this cell are organelles. We have the organs as a human, but in the cell there are no organs. They contain the organelles which do specific function. Okay, which do the specific function. So organelles are inside of the cytoplasm. So this is the liquid portion, and in this liquid we have the organelles. All organelles in the nucleus and in the mitochondria, this is the nucleus, okay? And these structures are called the mitochondria. In the nucleus and in the mitochondria, we have two membranes. We have two membranes, okay? Not one membrane. In the cell, we have the one membrane, but all organelles also contain the membrane. They separate them from the cytoplasm. In the nucleus and in the mitochondrion, we have two membranes, okay? This is very important. We have two membranes. But in the plant cell, the chloroplasts, do you know what are the chloroplasts?
It's a green color.
Yes, and it's also organoid. In the chloroplasts of the plant cells, they also have two membranes, okay? They also have two membranes. But in animal cells, how many chloroplasts we have? In human body, how many chloroplasts we have?
We don't
We
have.
don't have.
Yes, we don't have. So we only have nucleus and mitochondria, which has two membranes. But in cells, if I ask the cells how many organoids in the plant cell contains two membranes, you have to answer first of all nucleus, then mitochondria, then chloroplasts. Okay. And all other organelles have only one membrane, but in the ribosome, these tiny structures, they are called the ribosomes, these tiny structures, they are the ribosomes. In those ribosomes, what's the function of the ribosome? Who knows? Function of the ribosome, come on. Protein synthesis. Protein synthesis, yes. In the ribosomes, we have no membranes. No membranes, okay? No membranes in ribosomes. So we have a table. We have the table of the organoids. We are going to learn first, second, third, fourth, fifth, sixth, seven, 8, 9, and 10. Okay, we should learn them by heart. We should learn them by heart. Everyone should learn them by heart. Okay, so vacuole. Vacuole is in which cell? The vacuole contains in which cells? Have you ever heard about the vacuole? Which cell contain the vacuole?
Plants and animals.
plants and the animals, they contain the vacuole, mostly the plants, right? So they store the water in plant cells, okay? The vacuoles are going to store the water, storage of the wastes in animals and in the plant cells and other materials. For example, the water is going to be contained in the plant cells because in the plants we have the leaves, um, we have the body parts, right? All of them contains the water. If there is no water, they're going to dry. So vacuoles are the main structures to make actually to make stable the plant cells, okay, mostly. So ribosomes are places where the protein synthesis occur. This is the site of protein synthesis, and there is no membrane in the ribosomes. You have to know it, no membrane, okay, not bound by membrane. So smooth endoplasmic reticulum. What's smooth endoplasmic reticulum? This is the system of the membranes. It is the system of the membranes and the tubes that transport substances around the cell. So let's look first of all to ribosomes. These structures are ribosomes, and ribosomes. Their main function is protein synthesis. Okay, so they're going to synthesize the proteins. And after that, we have the smooth endoplasmic reticulum. This structure, this is a lightish yellowish structure, is called smooth endoplasmic reticulum. And here you can see the darker inside, the darker greenish color. This is what? This is rough endoplasmic reticulum. So this is the rough endoplasmic reticulum, which is closer to the nucleus. This is the nucleus of the cell. And if you exit the nucleus, you start to see the rough endoplasmic reticulum. And this is the smooth endoplasmic reticulum. So there are tubes and cisternas. There are tubes in the where in smooth endoplasmic reticulum, their function, they are everywhere. You can see them everywhere. So their main function is to transport the proteins or substances around the cell. It's like a logistics center in our body, okay? It's going to deliver the substances. They're going to it's written here, they're going to transport the substances around the cell. Okay, this is a function of the smooth endoplasmic reticulum. And they're going to synthesize the lipids, plus they're going to synthesize the lipids. And we have the rough endoplasmic reticulum. In the rough endoplasmic reticulum, what can you see in the rough endoplasmic reticulum on the surface? What you see on the surface of the rough endoplasmic reticulum?
Ribosomes.
Ribosomes.
Yes, we can see the ribosomes. That's why they are called, they are called rough. What's the meaning of rough? Bujmaygan, right? Bujmaygan. Sharahavata. What's the meaning of the smooth? They are smooth endoplasmic reticulum. They are rough endoplasmic reticulum. Smooth means gladka, silk. This one is Hadar budur sharahavata. Okay. So what's the function of the rough endoplasmic reticulum? So rough endoplasmic reticulum is a type. It's a type of the endoplasmic reticulum with the ribosomes. They contain the ribosomes. Okay, and those ribosomes are sitting on endoplasmic reticulum. So ribosomes on the rough ER means here endoplasmic reticulum, okay? Plasmic means here endoplasmic reticulum. So this endoplasmic reticulum, Golgi apparatus. What's Golgi apparatus? Who knows? Golgi apparatus. So here we have the ribosomes, right? Here we have the ribosomes. Here we have the ribosomes, and those ribosomes are going to synthesize the proteins. They're going to synthesize special proteins, which we will go to the membrane. So they secrete the transport, they're going to secrete the carrier proteins, channel proteins, okay? The ribosomes which are sitting on the surface of rough endoplasmic reticulum. And we have the Golgi apparatus. This is the Golgi body. This is called the Golgi apparatus, this bluish or purple in color. This is Golgi apparatus. So what its function? Its function is to sort and package proteins made by ribosomes on the rough endoplasmic reticulum. So what does it mean? So in the rough endoplasmic reticulum, the ribosomes are going to synthesize the proteins. Then those proteins are going to go to the Golgi apparatus. In the Golgi apparatus, they're going to be sorted and packaged. Okay, they sorted and package it, and after that it goes to the smooth endoplasmic reticulum and go throughout the cell. Throughout the cell, it goes to the whole cell. Throughout the cell, you have to know this. And what else? Sorts and packaged proteins made by the ribosomes on the rough endoplasmic reticulum. So mitochondria, what's mitochondria? Who knows?
Mitochondria provides the cell with energy.
Yes, mitochondria, first of all, takes part in the cellular respiration. It takes part in the cellular respiration. They're going to make the ATP, adenosine triphosphate. This is the energy for the cell. And the third characteristics of the mitochondria is that it's bound by two membranes. It has two membranes. Which organoid also has two membranes?
Nucleus and
Nuclear.
Chloroplasts in the plant cells. Okay, thank you. Good. So mitochondria, you have to tell. If I ask about you, what's mitochondria, you have to say first function in the cellular respiration. They're going to produce the ATP for the cell, and they're bound by double membrane. Okay. And we have the lysosomes. What's the function of the lysosomes? Here you can see this is the lysosome molecule. Its main function is to kill the bacteria and viruses, right? To kill the pathogenic all cells. And also we have the old cells inside of the cell. We have the old organelles inside of the cell. They're going to warn them out. They're going to release it outside. They're going to, first of all, digest, because in the lysosomes, we have the digestive enzymes inside. Digestive enzymes. Enzymes means fermente. Through these digestive enzymes, they're going to kill the bacterial viruses and also the old organelles, and they're going to warn out them outside. Okay, this is a function of the lysosome. And don't touch to the centrioles yet. We don't need the centrioles for now. We will learn during the mitosis. So we have the nucleus. What's the function of the nucleus? Who knows?
It contains chromosomes and DNA.
Yes, it contains the genetic information, which is called the chromosome, right? It contains the chromosome. So what's going to happen to that chromosome? It contains the DNA. So the main function of nucleus is to contain the genetic information, our genetic code, which is called the DNA. And after that, this nucleus is a control center of the cell. It controls everything which is happening inside of the cell. Everything is controlled by the nucleus. And also, this nucleus also has how many membranes?
Two.
Two.
This nucleus has two membranes. Yes, exactly right. It has two uh, membranes in this nucleus. And inside of the nucleus, we have a structure which is smaller structure, which is called the nucleolus, yadrishka, yoki nima deydi o'zbekchasiga? Yadrocha deyiladi, to'g'rimi? Nucleolus. What's the function of the nucleolus? Who knows?
For synthesizing RNA.
And
Ribosome assembly.
yes, great. They're going to synthesize the ribosomes and RNA. It's going to synthesize the ribosome and RNA. You're right. So nucleolus, they are small and dense structures inside of the nucleus. They are the sites for the ribosome synthesis and RNA synthesis. Okay, you should know it, ribosome synthesis. And the last organelle that you have to remember is the cell membrane. So cell membrane This is the outer membrane that regulates what comes into the cell and what goes out of the cell. It does the active diffusion, passive diffusion, facilitated diffusion, bulk transport. Everything is done by the cell membrane. Okay, good. Any questions regarding the cell organelles?
No.
Any questions? And in the last in the next lecture, I am going to ask one by one. For example, Nurhayot, I will give you a question. Nurhayot, can you please tell me 10 organelles with their functions? You're going to start with vacuole, ribosomes, then endoplasmic, RAF with their functions. Golgi apparatus, mitochondria, lysosomes, nucleus, nucleolus, cell membrane, everything. Everything should be mentioned, okay, in the next lecture. Okay, Saudat, did you understand? Munisa,
Yes.
Yes.
Kulhayo?
Yes.
Okay, good. Any questions regarding it?
No.
Okay. So that was, there is a video you can watch, the explanation of the organelles again, okay, if you didn't understand anything, but it's in Uzbek language, okay? And then after that, will we do this today or next lecture? What do you think? Will we do this exercise today or next lecture? This is matching. Can you do it now?
Yes.
Okay, I give you then 5 minutes. You have to finish it, okay? And we will discuss each of you. I will ask from each of you, okay? Good luck.
Can I do it the next lesson?
So that please do try, okay? Try to do it.